Welcome you to Violet Kovacheva's PhD viva talk, and my special thanks go to the examiners, Carolina Welby from Uppsala and Sarah Calvara. And in keeping with the rules, please refrain from asking any questions during the talk or after the talk. We leave that to the examiners. And without further ado, I'd like to announce Violet's talk on modeling and analysis of the tumor microenvironment of colorectal cancer. Please.
Hello everyone, and thank you for coming. So yes, as Till said, my PhD was on modeling and analysis of the tumor microenvironment of colorectal cancer. First, just to give you a brief outline, so I'll first talk to you about some of the background of my research and the motivations behind it. I will then introduce a method for cell-level protein network analysis for multiplex immunofluorescence, and then I'll talk to you about a model I have developed for the spatial tumor heterogeneity in the colorectal tissue, and how I have extended this to model protein expression. So what is the tumor microenvironment? Recently, so over the last few years, the view of the tumors have shifted from a disease that involves only the epithelial cells to a multicellular disease that really involves not only the epithelial cells, but also the surrounding cells. So the tumor microenvironment, it refers to the various types of cells, soluble factors, signaling molecules, and the extracellular matrix that surrounds the tumor cells. And it has actually been shown to promote tumor growth, invasion, and protect the tumor from the immune host system. So the tumor microenvironment is quite a complex and dynamic system, and in order to understand it, we need to look into it from various points of view. So we need to consider the protein co-expressions, and that also involves the interactions between different cells in the tissue. We also need to look at the architecture or the spatial tumor microenvironment, how that changes as the tumor progresses, and also the heterogeneity or the differences that we observe both within the tumor and between different tumors. So my research has focused on colorectal cancer. The reason for that is that colon cancer accounts for about 10% of all cancers, but it's actually the fourth leading cause of death in the world from cancer. Every year around 1 million people will develop it, and unfortunately about 50% of those will die within the 5 years from diagnosis. And the current way of staging it, the tumor node metastasis staging system, is actually a very bad predictor. It can tell us very little as to what will happen to the patient, what the outcomes and best treatments are. So for those of you who are not familiar with the colon tissue, this is what the healthy architecture looks like. So we have these glandular structures called crypts. These are made up of epithelial goblet cells with lumen in the middle, and this is where water would be absorbed in the colon. And then in between there's this connective tissue called stroma, and this is made up of various types of cells, including the different types of immune cells. So the first point of view that I take was look into multiplex protein imaging. So because of the complexity of the cancer, really no single marker is actually specific enough or sensitive enough to give us a full diagnosis for the patient. So multiplex imaging actually allows us to assess the complexity of the tumor microenvironment and the heterogeneity of the cells. It also provides a way for us to analyze individual cells, cell phenotypes, and the localized protein networks within anatomically intact tissue. So over the last few years, there have been several techniques proposed for multiplex imaging. These involve things like MODI, MXIF proposed by GE, and a couple of image mass cytometry methods. We have used the toponym imaging system, or TIS, as there is one installed here in Warwick. And this is basically a robotically controlled cyclic multi-channel fluorescence microscope. So the way it works is that we start with a single tissue sample, we wash and bleach this to remove any autofluorescence as much as we can from the sample. We then take a phase and a fluorescence image and incubate with an antibody and then take another set of phase and fluorescent images. So the phase images capture the structure of the tissue, they don't change between runs, so we use these to register the images afterwards. The fluorescence image before incubation captures any autofluorescence so that we can then easily remove this from the analysis. And of course the second fluorescence image captures the protein expression. And then we repeat the cycle, so we wash and bleach the previous antibody and do the same thing with all the antibodies that we're interested in imaging. So just to give you an idea of the variation that we can see in the protein expressions, so here we have a cancer sample at the top and a healthy sample at the bottom, and here you can see these correct structures that I mentioned earlier. So the first column here shows the dapper tag that stains the chromatin, so it allows us to identify the nuclear regions and hence segment the cells. With that in mind, the problem that we were now facing is, given this test data, how we can find subsets of proteins that are interacting in one type of tissue, one type of patient, and not in the others. For this I have proposed a framework which starts with inputting the fluorescence and phase images. These are pre-processed to remove any autofluorescence or artifacts within the tissue, and as I mentioned, we use the phase images to align the fluorescence images. We then use the Dagger channel to segment the nuclei so we can get all the regions that correspond to cells. And we then calculate the protein-protein dependence profile for each cell. So we have done this using a measure called the maximum information coefficient, which has been shown to be able to capture dependencies between data that are both functional and non-functional, and is very robust to noise in the data. According to this profile, then we phenotype the cells, and for this I have tried several different clustering methods, and I have actually shown that the final results I obtain are reasonably independent on the phenotyping method that we have chosen. I then calculate the DSWAP measure, and I'll tell you a bit more about this in a second. And then I output the social network of proteins for the proteins that we have imaged within the samples that we've analyzed. So what is DSWAP? So DSWAP stands for the difference in sums of weighted codependence profiles. It considers the protein-protein dependence value for a protein pair and normalizes this by the frequency of the phenotype within the sample. We only consider the highest values for protein-protein dependence and the most frequent clusters within each sample. And we sum up all the occurrences within cancer samples, the normal samples. We normalize by the number of samples that we have in each group, and we look at the difference. So here a positive score would identify protein pair which is more codependent, and this codependence occurs more often within the cancer samples. And respectively, if we have a negative score, it means that the protein pair are more likely to interact within the normal samples, and possibly this interaction breaks down in the cancer. So here are some of the final results. So I have shown you two different clustering methods, and these are mostly chosen because of their very different approach to clustering. And within these graphs, I have actually looked at only the extreme values. So the fact that some of the lines may not be present in one of the graphs doesn't necessarily mean that no interaction was found. It means that it sort of didn't make the cut. It wasn't one of the extreme values that I wanted to look at. But what we can see, we have these pairs, CA and EPCAM, and this consistently came with a very high value, so the protein pair is more likely to interact within the cancer samples. And then we have this pair CD36 and CD57, which interacts more often within the normal samples. So in order to analyze these results a little bit further, we went and looked into some protein interaction databases. And we found that actually this pair, CEA and NEPCAM, has been found experimentally to have a pathway through Claudin 7 and SOX9, which is specific to colon cancer. So it's an important pathway within the tumorigenesis. I also wanted to actually look back into the data and see where are these cells that I'm identifying which have these high interaction levels. So I've developed this tool which allows us to look at several samples at the same time. We look at cells which are expressing specified protein pairs, so I have here CEA and EpCAM, and cells that have high dependence between those two profiles, so above a set threshold, are shown in red here. And then we also look at the protein pair CD36 and CD57, and the cells that show high interaction for these two proteins are shown in green with overlap in yellow. And you can see, so here we have two cancerous samples. We can see a lot more red cells, whereas the two normal samples over there, we get a lot more green, so a lot more of these CD36, CD57 cells. So this allows us to actually look at the variation within each sample, so look at the different phenotypes of cells that are present, and also compare the different samples, and so look at the intertumor heterogeneity. So the other point of view that I wanted to look at is look at the spatial microenvironment, and I have done this by modeling the architecture of the colon. So I developed this model which takes a number of user-defined inputs. So these include the cell grade, the cell overlap and cellularity, so that is the density of the cells within the sample, also the image resolution, noise variance for the deterioration of the image. We also performed detailed analysis of hand-marked histology images, and from these we extracted various features to make the model more realistic. And these included things like the nuclear size, the nuclear textures, the cell phenotypes that are present within the tissues, also the crypt architecture, lumen texture, etc. So the way the model works is that we start by first generating the overall architecture, so choose the crypt locations, the crypt shapes and sizes, construct the lumen. We then go on to generate each cell individually, and then finally these ideal images are deteriorated to reflect what happens from the optical system. We then output a final image, and we did extend the model into also generating histology images. And this is because this type of images is much more commonly used within the clinical practice. The pathologists are much more comfortable in evaluating the appearance and working with these sort of images, and also it greatly improves the visualization of the model. So we also obtain a number of ground truth data, such as the cell segmentation, crypt segmentation, stain normalization, etc. So I showed you what the healthy tissue looks like. This is what the synthetic looks like. So this is what the model can output. With well-differentiated cases, so this is the slightly cancerous, sort of less malignant ones. What happens is that we still have these glandular structures, but their shape is a bit more deformed. We lose the goblet cell structures, and this is what I've tried to reflect in the model. For moderately differentiated, so the more malignant cases, things get progressively worse, so the crypts become more distorted in shape. We can see that it's very difficult to actually identify individual crypt structures. Here we also have some necrosis, so dead cells within this crypt. And actually it's worth pointing out here that these textures within the crypt that you can see there, I've actually put in after submitting my thesis. So this was a part of an improvement on the model for a journal paper that I'm preparing. And then lastly, for the poorly differentiated cases, what happens within the real data is that there are now very few crypts that we can observe. Their structure can be very complicated. What we tend to see more is clusters or sheets of cancer cells. And this is again what I've tried to mimic within the model. So to evaluate the appearance of the model and the synthetic data, we asked 3 pathologists to grade how realistic various features appear. And you can see that actually they graded things quite highly in general, with the only exception being the stromal cells. And for that they said they could identify what types of cells they were. They could identify that they are stromal cells, sorry, but they couldn't identify what precise type of cells they were. And also another way of validating the model was, so because the nuclear texture is an important factor when grading this type of tissue, we wanted to make sure that it actually is very realistic. So we generated a set of synthetic images and compared that together with real images, and I've compared the nuclear texture based on the heraldic texture features, clustered these together. And we can see, for one, the clustering method was not able to distinguish between the 2 types of data. We always had clusters that included both real and synthetic data, but also we have actually reasonably even distributions of the 2 data within the samples generated. In addition to this, since the crypts are the main architectural features, we want to see how realistic they are. So we've applied a crypt segmentation method using different test and training sets. So we did several experiments, and you can see when we trained the algorithm on synthetic data and tested on synthetic data, it performed quite well, and the results were actually closely comparable to what we see with real data when different sets are compared. When training the segmentation algorithm on real data and testing it on synthetic data, the algorithm performed quite well for healthy and well-differentiated cases, but initially it performed quite poorly for the high-grade cancers. And here in blue are things that I've done after submission of the thesis. So this was after the introduction of this crypt texture. And you can see actually that resulted in a very significant improvement in the results. I have now also looked into what happens if I train the algorithm on synthetic data and test it on real data, and we can see actually I obtained quite reasonable, quite good results. Lastly, I wanted to see what happens if I try to combine the 2 strands. So for this I've looked into expanding the model to include models of protein expression and hence generate multiplex fluorescence data. For this I've generated now models for 6 proteins, namely MLH1, PMS2, MSH2 and MSH6, P53, and I've now included P10 as well. And these are important for studying microsatellite instability, which can have implications for the treatment of the patient, and so is commonly screened within the clinical practice. I wanted to generate subcellular expression models, and for this I also needed to include models for the nucleoli, Golgi and vesicles. So these are the protein localization patterns that are known, and these were obtained from the Human Protein Atlas database. And you can see we have quite a wide variety of expression patterns. And also, as I mentioned, so these proteins were chosen as a case study in order to demonstrate how proteins can generally be added. So we can easily, in the same way, add a lot more proteins to the model. So, as I mentioned, as the first step, we needed to generate cell organelles models. So from this we performed detailed analysis of high-resolution confocal images from the Human Protein Atlas. We extracted various features and we have embedded these, the distributions of these within the model. And here I've shown you a few examples of how these organelles were generated, so for the 3 types of organelles. Lastly, so what the model actually generated. So we have here, as I mentioned, these high-resolution real confocal images that show you the subcellular location of the proteins of interest. I've also looked into which cells actually within the tissue express the proteins. So for these three proteins, if there is no mutation, then both epithelial and stromal cells will express it. When a mutation in one of these proteins occurs, then the epithelial cells within the tumor will not express this. In the end, I've shown you the results, and this is within a healthy sample, so there's no mutation, hence all cells express it. And I hope you would agree the expression patterns are reasonably similar, at least. And then another couple of examples. We have MSH2 and P53. With MSH2, again, it actually is expressed in all stromal cells, and we can see that as well in the simulated data. Whereas with P53, we actually see expression only in some of the epithelial cells, and this is what happens within the synthetic data. So here, only a couple of the epithelial cells are expressing this protein. And finally, I wanted to demonstrate how the DSWAP framework can be applied to this synthetic multiplex fluorescence data. So I compared images that do not have any mutation within the samples to images that have a mutation either in MLH1 or MSH2. And these mutations, as I said, result in the epithelial cells not expressing these. Actually what we found was that the DSWAP measure was able to identify the mutations. And however, with these particular proteins, actually just looking at the protein expression values would still be able to identify the mutations. And really the value of the DSWAP measure actually lies with proteins that change subcellular location in the different types of tissue, which can actually easily be implemented within the model. It's just the proteins we've chosen are not known to change location. So, in conclusions, I've studied the tumor heterogeneity of colon tissue. I've introduced a method for analyzing multiplex immunofluorescence, which highlights protein pairs which have different interaction patterns within healthy and cancerous tissue. I have also proposed a model for simulating both healthy and cancerous colon crypt architecture, and I have extended this to include protein expression models for a number of proteins. The generated data from these models can actually be used to validate a number of image analysis algorithms, such as crypt and cell segmentation, image restoration, cancer grading, stain normalization, etc. So finally, just a list of publications. So as I mentioned that I'm currently preparing a journal article on the model. I've also have submitted a conference paper that I'm waiting to hear from quite soon, and then the other published papers. And I would just like to thank my supervisor, Dr. Nasir Rajput, our other collaborators, and everyone in my group. And thank you for listening.